Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk on the module Biosynthesis of Lipids Part 7 from the paper Lipid Metabolism. The objectives of the module includes understanding the biosynthesis of glycerol, which is an integral part of the lipid, the significance and the implications of the synthesis of glycerol, as well as its application. Glycerol, synonymously known as glycerine or glycerine, is a simple polyol compound, or else we can say it's a sugar alcohol. Glycerol is the backbone of triglycerides or the triacylglycerol, and both the glycerol as well as fatty acid that forms triglycerides are crucial to all lipids. Glycerol is a viscous, odorless, colorless, non-toxic liquid. It is sweet in taste and extensively used in many pharmaceutical preparations and formulations. Being hygroscopic in nature, glycerol has 3 hydroxyl groups conscientious for its water soluble. Glycerol is produced by the transesterification or the hydrolysis or the saponification of the triacylglycerols or triglycerides. It is also generated as a co-product in the assembly of long chain carboxylate salts, which are utilized as soaps or as a byproduct in the biodiesel productions. Glycerol is either derived from animals, for example tallow, or plants, for example palm or soybean, or maybe chemically synthesized. Looking at the broader perspective, the glycerol may either be obtained from the triacyl glycerols or the triglycerides, which is a natural form, or from the non-triglyceride source, which is a synthetic form. The crude glycerol from the triglycerides or the triacyl glycerols is burnt for energy due to its low heat value and hence dispose of as waste. Purified by activated carbon treatment to eliminate organic impurities, or by alkali treatment to eradicate the unreacted glycerol esters, or the glycerol may be purified by ion exchange chromatography to get rid of salts. Vacuum technique is also found useful for the purification of glycerol owing to its high boiling point, which is 290 degrees Celsius. In addition, the multi-step distillation process is also used to get high purity glycerol, which is more than 99.5% pure. Takes place when glycogen is broken down to form glucose. Then glucose through the glycolytic pathway forms glucose 6-phosphate, glucose fructose 6-phosphate, then fructose 1,6-phosphate, which further converted to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. From dihydroxyacetone phosphate, glycerol synthesis takes place. Whereas glyceraldehyde 3-phosphate also give rise to pyruvate and acetyl-CoA, finally to yield fatty acid, and these fatty acid as well as the glycerol synthesized from dihydroxyacetone phosphate together form lipids. Glycerol also can be convertible to dihydroxyacetone phosphate and further to 1-acyl dihydroxyacetone phosphate to give rise to lysophosphatidic acid. Through another pathway, glycerol give rise to glycerol 3-phosphate, and which also give rise to lysophosphatidic acid. This lysophosphatidic acid further gets converted to phosphatidic acid, and phosphatidic acid to diacylglycerol, and then diacylglycerol to triacylglycerol. The synthetic glycerol is produced by different routes from propylene. The steps of the synthesis of glycerol can be: the first is the chlorination of propylene. The most vital is epichlorohydrin process, which yield allyl chloride followed by its oxidation with hypochlorite to get dichlorohydrins. Dichlorohydrins reacts with a strong base to provide epichlorohydrin, which is hydrolyzed to yield glycerol. The synthesis of glycerol can also be achieved from propylene oxide and acrolein. Coming to the applications of glycerol in food and beverages, glycerol dole out as a sweetener, humectants, and help in the preservation process. In low-fat foods, for example cookies, glycerol acts as a filler, and in liquors work as a thickening agent. Together with water, glycerol used to safeguard certain kinds of plant leaves. It is also used as a sugar substitute, providing 27 kilocalories per teaspoon. It also acts as an additive sweetener, and it is supplementary to icing to avert it from hard setting. In medical and personal care preparations, glycerol is mainly used as a humectant, providing lubrication and improve smoothness with its moisturizing assets. It is endowed in toothpaste, soaps, skincare products, shaving creams, mouthwashes, hair care products, expectorants, elixirs, and cough syrups. As a holding agent, glycerol is used in solid dosage forms such as tablets, and as a laxative agent, glycerol persuades a hyperosmotic effect and also found useful for reducing high eye pressure. 10% of glycerol averts the precipitation of tannins in the ethanol extracts of plant prepared as tinctures, and also used as a solvent substitute to ethanol in organizing herbal extraction free from alcohol. It has also been revealed that glycerol acquire a high degree of extractive versatility for botanicals as well as elimination of several constituents and intricate compounds. It also possesses the inherent assets of rendering or not denaturing an inert constituent of botanicals and is an unwavering preserving agent for extracts. Glycerol is bacteriostatic in its action and is an element of electronic cigarette solution as a e-liquid. Glycerol is a non-ionic cosmotrope similar to that of propylene glycol and ethylene glycol, and with water molecules, glycerol forms Brownian hydrogen bonds. It was historically used as an antifreeze agent for automotive applications prior to its replacement by ethylene glycol, which lowers the freezing point. Glycerol is also exploited as a cryoprotectant for microbial culture, for cell lines, and for embryo storage. It is also used to power diesel generators, furnishing electricity in nitroglycerin production as an essential ingredient of cordite, dynamite, gelgenite, and also as an aerosol spray. Glycerol is also used in allyl iodide manufacturing, hydrogen gas production unit, glycerine acetate formation, acrolein, epichlorohydrin, propylene glycol, and ethanol conversion. Glycerol is also used in cinema to impede the areas drying out too quickly. As far as the metabolism of glycerol is concerned, the synthesis of glycerol starts with the breakdown of the glycogen, its conversion to glucose 1-phosphate by phosphorylase enzyme, and then further conversion to glucose 6-phosphate by mutase enzyme results in the formation of fructose 6-phosphate and fructose 6,1,6-diphosphate with the utilization of ATP, dihydroxyacetone, as well as glyceraldehyde 3-phosphate is formed. Dihydroxyacetone phosphate further transformed to glycerol 3-phosphate and finally lead to the formation of glycerol. In humans, glycerol, which is a 3-carbon naturally alkyl alcohol, is the structural backbone of triglycerides or triacylglycerol, which is transformed to a glycolytic substrate for ensuing metabolism. The synthesis of the glycerol takes place in the interconvertible steps, like glycogen from the liver is broken down into glucose 1-phosphate. This glucose 1-phosphate is also referred to as Corey's ester, and this conversion takes place by the catalytic action of glycogen phosphorylase enzyme. The glucose 1-phosphate further transformed to glucose 6-phosphate by phosphoglucomutase (PGM), which is an enzyme that transfers the phosphate group from first to sixth position or vice versa. Glucose 6-phosphate undergo change to form fructose 6-phosphate, which is a Newburgh ester, by phosphoglucose isomerase enzyme. The phosphofructokinase enzyme transformed the fructose 6-phosphate to fructose 1,6-biphosphate, which is hardened young ester. Now, fructose 1,6-bisphosphate is broken down into dihydroxyacetone phosphate as well as in glyceraldehyde 3-phosphate by phosphofructose bisphosphate aldolase. Then further, dihydroxyacetone phosphate get reduced to glyceraldehyde 3-phosphate by glycerol 3-phosphate dehydrogenase, and this step endows the adipose tissue to get triggered glycerol backbone for the synthesis of new triacylglycerols or the triglycerides. The reaction requires either NAD plus or NADH as a cofactor. In Leishmania mexicana, which is a protozoan parasite, dihydroxyacetone phosphate also has a function in ether lipid biosynthesis. Finally, glycerol 3-phosphates gets converted to glycerol. In the de novo synthesis of glycerolipids, glycerol 3-phosphate is the preparatory material and is the foremost acylated on its SN1 position by glycerol 3-phosphate O-acyl transferase, which is a mitochondrial endoplasmic reticulum membrane enzyme. Further, acyl group is supplemented on the SN2 position, assembling the synthesis of phosphatidic acids. In several fungi, glycerol 1-phosphate is present, and through the catalytic action of phosphatase, the phosphate moiety of glycerol 3-phosphate produces glycerol. In addition, they can also execute fermentation of glycerol by fabricating glycerol from glucose via glycolysis pathway. The glycerol 1-phosphate, occasionally called as D-glycerol 3-phosphate, is an enantiomer of glycerol 3-phosphate. A majority of organisms utilize L configuration or 3-phosphates for glycerolipid backbone, though 1-phosphate is explicitly employed in the ether lipid of archaea. In adipose tissue and liver, glycerol is a precursor for the synthesis of phospholipids and triacylglycerols. The breakdown of the fats or lipids results in the release of fatty acids and glycerol in the bloodstream, and the glycerol moiety neither glycate proteins nor do guide to the advanced glycation end product formation. Conversely, in various organisms, the glycerol enters the glycolytic pathway or the glycolysis unservingly, and accordingly affords energy for the cellular metabolism. Before entering into the glycolytic and or gluconeogenesis pathway, glycerol ought to be transformed to their transitional glyceraldehyde 3-phosphate. The steps of the conversion is depicted below. In the first step, the glycerol gets converted to glycerol 3-phosphate by glycerol kinase. The enzyme glycerol kinase exists in brain, in kidneys, in liver, in muscle, and in other body tissues. The next step, the so formed glycerol 3-phosphate undergoes transformation to form dihydroxyacetone phosphate by the catalytic action of enzyme glycerol 3-phosphate dehydrogenase. Furthermore, the dihydroxyacetone phosphate is interconvertible to glyceraldehyde 3-phosphate by the catalytic action of triose phosphate isomerase. Glycerol, when ingested, flaunts a very low toxicity. In small animals, for example, mice and rats, the reported lethal dose at 50% of oral dose is 8,700 milligram per kilogram, and $12,600 milligram per kilogram respectively. In 2007, the United States Food and Drug Administration instructed all medicine makers to check all the batches of glycerol for the toxic diethylene glycol that followed the fatal cases of poisoning in Panama resulted from the falsified import customs declarations. The contemptible diethylene glycol was then labeled as extra costly glycerol. So, today in this lecture, we have learned about the biosynthesis of glycerol, how glycerol is synthesized from the glycogen moiety, as well as the breakdown of the triacyl glycerol resulting in formation of glycerol. We have also come to know about the applications of the glycerol in various formulations and preparations of pharmaceutical significance, and the interplay between the glycerol as well as glycolysis pathway. Thank you.